saw it, we could hardly believe it.
As a scientist, it was very surprising.
So when the results came in, I was like, wow.
When I saw the data, I couldn't believe them.
This was completely unexpected.
February 2012. Researchers at the prestigious North Carolina Research Campus Human Performance Laboratory make a startling discovery. One that answers some important questions and opens the door to even more. ASEA is the world's first and only redox signaling supplement. Redox signaling works on a cellular level, and its primary functions ensure the ongoing vitality of our cells, including proper cell metabolism. Metabolism is the name we give to the chemical reactions that take place inside our cells in order to sustain life. When these chemical reactions happen, they leave behind traces, almost like fingerprints. We call these fingerprints metabolites. One of the premier institutions for studying metabolites and their importance to human health is at the prestigious North Carolina Research Campus Human Performance Laboratory. And so, ASEA contacted Dr. David C. Neiman, director of the Human Performance Laboratory at Appalachian State University. Dr. Neiman, the vice president of the American College of Sports Medicine, is the author of 9 books dealing with physiology and exercise testing, more than 200 peer-reviewed publications, and more than 300 articles in health and wellness magazines.
We have a national reputation for testing various sports nutrition products and their effect on performance and exercise-induced physiologic stress. So this campus, the North Carolina Research Campus, we have several universities that are here, including Duke, UNC Chapel Hill, NC State University, and we're working together with various companies, Dole Foods, General Mills, and we're all working here together really under one mission, which is the connection between nutrition and health and performance.
And so in late 2011, Dr. Neiman and several other PhDs carefully crafted a scientific study. A study designed to measure the effect of ASEA on metabolites, the fingerprints left behind as a result of cell metabolism.
So the way that we conducted the trial was to randomly assign half of the subjects to one treatment and half of the subjects to the other treatment. It's a double-blinded study, meaning that we as the investigators had no idea who we were assigning to what condition, an A condition and a B condition, but we didn't know what those were. So the subjects are assigned to either A or B. They come in and they do the first trial and they ride as hard as they possibly can, and then they cross over, meaning that if they were on condition A to start with, they would then transition to treatment B or condition B for the second trial, and vice versa, B to A. And so in that way, we're really testing, we're taking everything out of the equation as best we can by just leaving it to the athlete and the bike and riding as hard as they possibly can to test the possible benefit of ASEA.
We drew blood samples on the athletes after they had taken ASEA or placebo for a week. That was just before they cycled 75 kilometers, and then we took another blood sample after that, and then an hour later.
The rigorous protocols of the double-blinded crossover study took several weeks. The ASEA corporate team immediately flew to North Carolina, where they learned that the results of the study surprised the entire group of PhDs. They were expecting to see something when ASEA was combined with exercise. Instead, they found chronic results, that is, results that happened before exercise, simply from drinking ASEA.
We didn't think that drinking ASEA would shift metabolites chronically. We thought it would do something during exercise, but not after a week of drinking it. After working with the bioinformatics statistical division, we were able to determine that drinking ASEA over 1 week caused a shift in 43 metabolites. Not a little shift, it was a large shift that caught us by surprise.
We have rarely seen such a drastic difference. In the compounds that we look at, we're looking at probably about 150 to 200 compounds. For 43 of those signals to change, that's a quarter of the metabolite profile that we're monitoring. So it's a huge bump in metabolite shifts due to just ingestion of one product. So it's a drastic difference.
I didn't believe it. So when I saw this, that 43 metabolites were differentially, they were at different levels. I didn't believe it. And I've done studies with microarray analysis where you're looking at thousands and thousands of genes, and you might find 2 or 3 genes that are differentially expressed. But here we are in metabolomics, and you find 43 things that are different. It was astounding.
The data revealed shifts in 43 metabolites, something these PhDs had never seen before, something that caused these scientists to use words like astounding and massive. But what exactly did these metabolite shifts mean?
Many of the metabolites were related to plasma-free fatty acids. So the athletes actually started the exercise after drinking ASEA with a lot more of these free fatty acids in their blood. The reason that's important is then the muscles will actually use that as fuel, sparing the muscle glycogen and the use of amino acids, which is what we found as we analyzed the data.
And the free fatty acids are very important because during exercise, we'll oxidize those to make energy in the form of ATP.
This is exciting news, and again, this research that we've seen that can prime the body to actually put you in a better position pre-exercise with this particular product. This is exciting news.
Drinking ASEA for a week caused this increase in free fatty acids. Every understanding from the literature is that these probably came from the fat stores in the abdominal area, and we'll be taking a closer look at that. So if you drink ASEA, we found that the fats go up in the blood. If you're not exercising, those fats will still be used to support the body's metabolism for life.
Metabolomics, the ideal tool for measuring the effect of supplementation on the human body, revealed astonishing properties of ASEA. Simply drinking ASEA for 7 days mobilized fuel stores in the body, fuel stores that almost certainly come from abdominal fat. For those who want to burn more fat during exercise, ASEA is ideal. Even without exercise, the freed up fat stores will be used by the body as fuel. Since this fuel source is available at the start of exercise, it's likely that muscle glycogen is spared, giving athletes who drink ASEA a huge advantage. And additional screenings showed that ASEA does all this with no negative effects on inflammation, oxidative stress, immunology, and diagnostic chemistries for liver and kidney function. This breakthrough discovery gives clarity and insight into one aspect of ASEA and its benefits. And it's just the beginning.
These are extremely important data, and we look forward to doing further research. These data we feel are very important and fascinating, and we intend to better understand what's in the drink and this shift in the metabolism that occurred. We want to understand what that means for every individual.
Redox signaling as a body of science is large, for sure. Probably larger than any of us even imagine. It's that humanity and the impact on humanity that underscores our full commitment to pushing the boundaries of understanding the science.